Hello everyone and welcome to today's webinar. On behalf of Drug Target Review and Eurofins DiscoverX products, I'd like to thank you all for attending. I am your moderator Izzy Wood, the editorial assistant at Drug Target Review. Our speakers today will be Dr. Gaurav Agrawal, who is the Assistant Director of Market Development at Eurofins DiscoverX, and Dr. Giulia Mancini, who is the Bioassay Lab Manager at Eurofins BioPharma Testing. Following their presentation, we will move on to a live question and answer session where you can pose any questions for today's speakers. Please remember you can submit questions at any point during the webinar using the questions panel situated on the menu on the right. So without any hesitation, I'm going to pass over to Gaurav and Giulia. Thank you.
Thank you for the introductions. Hi everyone, welcome to this webinar. Let's dive right in. We have a very focused presentation for you today. Here is an outline for it. In the first part of the presentation, I will take you through a brief introduction to Eurofins DiscoverX and an account on GLP-1 and GLP-2 as key metabolic targets. And before I hand off to Giulia, we'll give an overview of Eurofins DiscoverX's strategy behind building and qualifying phase-appropriate bioassay formats from stable cell lines. Giulia will then talk about Eurofins BioPharma product testing capabilities at Milan site and will present two case studies validating GLP-1 and GLP-2 bioassays at their facility. And working together, we ensure that companies who are developing GLP analogs for commercial release can rely on us to provide end-to-end support for accelerating their lot release testing programs. So let me start with a brief introduction to our company. Here's a bird's eye view, so to speak, of our core capabilities at Eurofins DiscoverX. We are a products division of Eurofins Discovery based in San Francisco Bay Area and are enabling drug discovery and development programs for over the last 20 years. We come with a deep experience in engineering novel MOA-reflective functional cell-based assays. In fact, we have the largest menu of cell-based assays across the industry, covering 10 plus major target classes in phase-appropriate fit-for-purpose formats, such as qualified ready-to-use bioassays, stable cell lines, and optimized assay reagents. We are closely working with biotech and biopharma companies implementing our assays in their drug discovery and QC lot release testing programs at their affiliated CROs and CMO sites, ensuring successful method transfer and assay implementation in a timely manner. The focus for today are GLP-1 and GLP-2. These are well-researched metabolically significant peptide hormones that are synthesized primarily in the L cells of the intestine. From here, they travel to various target organs, including kidneys, stomach, liver, pancreas, and the central nervous system to exert their desired metabolic effects. GLP-1 is crucial for glucose metabolism by increasing insulin secretion and downregulating glucagon. It also regulates appetite control by increasing satiety through central nervous system. It is now a well-established target for weight loss and diabetes management. While GLP-2 is a promising therapeutic target for inflammatory and short bowel syndrome and is demonstrated to play a protective role in reducing bone reabsorption, thereby increasing bone mass. GLP-2 and related analogs are emerging targets for the treatment of short bowel syndrome, Crohn's disease, and osteoporosis. Now, before I move further, here's our assay design. And I would like to quickly introduce you to the core technology that we use in several different ways to develop 1,000+ assay product portfolio. It's called enzyme fragment complementation technology, which is based on a split beta-galactosidase system, where the enzyme is split into two inactive fragments called ED or enzyme donor and the EA or the enzyme acceptor. Now, these fragments can be attached to different interacting proteins in the cell, and under the right biological stimuli, such as addition of an agonist ligand, a peptide, or a small molecule, the two proteins come together, bringing the two fragments to complement each other to form an active beta-gal enzyme. When added with the chemiluminescent substrate, the active functional enzyme hydrolyzes it to produce a light signal that can be read in any benchtop luminometer. On the bottom panel, highlighted are several distinct advantages, mainly that these assays are homogeneous and have a simple assay protocol. They don't require washing, shaking, or centrifugation or filtration steps. In addition, they produce robust assays with large signal-to-assay backgrounds. And in addition, the assays are designed with a simple and easy to implement protocol, making them easier to transfer between sites without steep learning curves for operators. Now, here's how we have used EFC technology in some very creative ways, building many different assay formats that represent the MOA and display accurate receptor pharmacology. Now, these are, for example, quantitating receptor dimerization, protein-protein interactions, protein turnover and degradation, secretion and intercellular trafficking. And in more recent years, we are developing assays for cytotoxicity applications such as ADCC and ADCP. Now, utilizing the EFC technology, we have built, in fact, the largest menu of cell-based assay portfolio in the industry with over 800 functional cell lines, and from that, hundreds of assay-ready kits and membrane preps. Our portfolio sufficiently covers all major druggable target classes, such as GPCRs, receptor and cytosolic tyrosine kinases, interleukins, checkpoint modulators, as well as reporter assays for key regulators of signaling pathway, and then cytotoxicity applications such as ADCC and ADCP. Now, focusing on glucose metabolism, listed here are the key targets that control glucose and other associated metabolic processes. Each of these targets here are of immense metabolic interest and has various clinically approved therapeutics in the market, and even more in clinical development. These includes the originator biologics, biosimilars, and a large number of peptide analogs. And here are some key examples of the drugs that are approved in the market. Now, in our effort to develop relevant assays that can be readily implemented in potency testing program, we have went an extra mile in qualifying our assays with these approved drugs shown on the slide to make them fit for purpose. In the coming slides, I will show you an example of such assay qualification work that we performed based on ICH guidelines. Now, moving on, let's look at the mechanism of action for the assays that we developed for these targets. As you can see, the receptors are either GPCRs or tyrosine receptor kinases. Now, for GPCRs, we have taken a more comprehensive approach and build assays with multiple different MOAs. The most common MOA recorded for the majority of GPCRs, as shown here, is the second messenger assay, quantifying cyclic AMP or calcium. Now, both of these signaling molecules are classical hallmarks representing GPCR activation and are accepted as true MOA. Next, we build assays recording the recruitment of beta-arrestin to the activated GPCR. Beta-arrestin recruitment is also a well-understood event and is also considered a proof of concept for a GPCR activation. Beta-arrestin assays are stoichiometric assays and present most accurate GPCR pharmacology. In addition, we created another assay format for GPCRs recording receptor internalization. It is well established that activated GPCRs undergo internalization upon ligand binding. Now, there are several ways that our customers are intelligently using this opportunity of having these multiple MOAs for the same target. Mainly looking at the phenomena such as ligand bias, where a ligand prefers to simulate one pathway over the other, leading to some very interesting pharmacological insights. And last but not the least, assays for receptor tyrosine kinases, such as insulin and IGF-1 shown here. And these are based on recruitment of SH2 domain containing proteins to the activated receptor. Now, recruitment of SH2 domain protein is among the very early events happening after activation. Recording such early events proximal to the site of action also helps us to root out any off-target effects, sometimes seen in gene reporter type assays. Now let's look at the assay formats. How do our customers access our assay products? For us, it is important to make sure that our assays are available in phase-appropriate assay formats. The primary is, of course, stable cell lines offering a continuous culture format. Our cell lines are passage stable and well characterized for functional performance. Now, from these cell lines, we have derived a ready-to-use format that essentially don't require any cell culture. The ready-to-use format is mindfully developed and optimized for as the most appropriate format for implementing in potency and stability testing programs. And finally, to demonstrate that the bioassays are fit for purpose, we further qualified them with clinically relevant marketed drugs. One such example I'll be showing in the following slides. The assay qualification was performed based on ICH guidelines that further enable these bioassays to be readily adopted for lot release testing. Next, let's quickly look at how our bioassays are configured. The upper panel here shows an overview of the bioassay kit itself. It comes with everything you will need to run the assay, including cryopreserved cells and all necessary reagents, and assay plates that are specifically optimized for the assay. And it comes with all necessary documentation, mainly a lot-specific certificate of analysis document showing the QC data for assay performance and data for mycoplasma and sterility testing, among other details. The lower panel shows a quick overview of the general assay protocol for our assays. The protocol remains similar across entire assay portfolio, and for this reason, it can be easy to implement and perfect assays in a new lab. So starting with plating cryopreserved cells directly into the assay plate, which is followed by addition of the molecule of interest. That could be an antibody, a ligand or peptide, or anything that stimulates the pathway. Now, upon incubation, the chemiluminescent signal detection reagent is added, and the plate can be read in any benchtop luminometer. Thus, the workflow doesn't require, as you see, any washing, shaking, or centrifugation step at all, thus making the assays very user-friendly. Now, adding some background to our strategy for bioassay development, it is important that we follow industry standards. We develop and qualify our bioassays in accordance with ICH guidelines to ensure that they are fit for purpose for implementation in QC lot release programs. Now, several attributes listed here are deemed essential in potency assays, but are often difficult to achieve and maintain over time. Mainly a high level of assay accuracy, precision, and reproducibility, which are essential for assays when performed on independent days, months, and years. Now, our assays accomplish this by implementing strict QC guidelines that bring high manufacturing consistency between lots. Now, in addition, we have in place several risk mitigation strategies for long-term support that enable us to maintain an uninterrupted supply of the qualified bioassays. So here I'm showing you a quick overview of typical assay qualification exercise that we performed. Here is a data sheet, data set for GLP-1 bioassay qualified with FDA-approved drugs. In this case, Ozempic, also commonly known as semaglutide. It's a GLP-1 agonist. So here's a table on the left. You see the data from the relative potency experiments across a wide range of 50 to 150%. The experiment here shows data from 2 individual analysts. Now, based on expected versus measured potency values across the range, the assay is deemed very accurate with 97% accuracy. A high degree of assay accuracy here suggests that the bioassay can measure relative potency with high confidence. Now, assay's intermediate precision was below 9%, which is excellent for assay repeatability when performed on different days and between 2 analysts. Also, the dilution linearity was analyzed by plotting the measured versus the expected relative potencies, and is shown in the figure on the top right. The R-square values came close to 99.9, showing an excellent assay linearity. Now, to bring all that we discussed in a unified perspective, the primary reason for a wide adoption to our qualified assay bioassay format is evident when we look at this typical roadmap for developing a fit-for-purpose bioassay for a biologic drug candidate, which is needed for its commercial release. It can take upwards of 18 months if starting from cell line development, subsequent qualification work, and implementation into QC lot release testing. Now the same figure looks quite different when we implement our qualified pre-validated bioassays, and this can really shrink this long time frame by over 14 months. Now, when implementing our qualified bioassays for the drug candidate, you will bypass a number of milestones that is on the top figure and can be looking at an assay that can be readily transferred to a GMP validation work. Like the qualified assay I showed you for Ozempic in a few slides before, here I'm showcasing some off-the-shelf bioassays that we have qualified for the mentioned biologic. All of them are FDA-approved biologics, and each of these assays is clinically relevant and is available off the shelf. And I will leave you here with this global overview of some of our most adopted assays that are currently implemented in various geographies for potency, stability, and NAB testing. At Eurofins DiscoverX, we are highly committed to providing end-to-end support for such biologic development programs, ensuring successful assay implementation at GMP CROs, sites just like BPT Milan. So thank you everyone for your time, and Julia, take it away.
So thank you, girls, for your nice presentation. Again, I'm Giulia Mancini. I'm the lab manager for bioassay testing here at Eurofins BPT Milan. Today, I will present some capabilities and validation approach which are used in our facility, and I will present two case studies, in particular, CMP assay validation for GLP-1 bioassay and beta-arrestin bioassay validation for GLP-2 bioassay, and some conclusions that are related to this very nice work together with our partners, which are Discovery. First of all, a brief presentation of our facility. Eurofins BioPharma Testing Milan deals with different testing activities from chemistry to molecular biology, covering the expertise in large and small molecules, medical device, and biocide testing. And overall extractable and leachable, including toxicology and also consultancy services. Here, the biologics business unit offers support to biopharmaceutical companies throughout the biologic life cycle, from analytical method development to market product release analysis. In particular, focused on the biochemistry and comprehensive expertise, including raw materials, drug substances, intermediates, and drug product testing. In addition, molecular biology and biosafety laboratories continuously develop different testing to support the life cycle of pharmaceutical products. And last but not least, the bioassay services, which cover R&D, optimization, validation according to GMP regulation, and with strategies based on the sponsor requirements, and as well as the international guidelines and recommendations such as ICH, FDA, ISO, and EMEA. In particular, on bioassay expertise, we can find a wide range of different biologic matrices. And for example, we can find monoclonal antibody, small molecules, peptides, vaccines, viral vectors, antibody drug conjugates, growth factor, scaffold proteins, peptide, and so on. And in this frame, it's really important to explain why the biologic product differs from conventional drugs. First of all, we have to underline the difference in size and complexity, and generally the different route of administration for the patient. They also have specific manufacturing processes and structural organization patterns. So to well characterize the biologics, the natural molecular heterogeneity, the impurity profile, and of course the potency need to be defined with a high degree of confidence. The characterization of these products has to be applied on the manufacturing process, product characterization and release testing. And to cover these three phases in the biologics business unit, is it possible to apply different techniques and testing? Here you can find just some example for the biochemistry side, in which you can find different techniques like liquid chromatography, gas chromatography, photometry, and so on. Going into molecular biology, we have the microbiology identification, mycoplasma testing, monocyte activation detection, genetic sequencing, and so on. And of course, in the bioassay testing, is it possible to test the mechanism of action and especially the potency of a drug? Of course, there are different applications that can be used, ranging from cell-based bioassay, immunoassay, ELISA, functional bioassay, enzymatic assay, different tests applied for cell culture, and so on. In Eurofins bioassay test in Italy, the activity are running under different approaches. First of all, research and development approach that usually is conducted in non-GXP environment, which means that all our facilities is GMP, but we can also decide to apply modification on the protocol, especially when we have a specific request for the clients that are running some molecules that are not in a clinical phase test. Of course, then we have the development phase, validation, or some transfer approaches. Today, we will focus on validation approach, and we will present a case study related to validation approaches. As I always said, its bioassay has its own story, and this is true. Indeed, there are some guidelines that it's possible to follow, for example, USP 1030, 1032, 1033, 34, and as well as the ICH Q2 R1. But to be honest, each bioassay and especially each molecules, each drugs has a different profile, a different story. So most important things is to have specific parameters panel tests and parameters that can be challenged during method validation, especially for quantitative bioassay testing. And we will present some data related to the bioassay quantitative test. For sure, it is necessary to set the suitability system test criteria, and then it's important to challenge the parameter of specificity, accuracy, precision, both for system and method precision, and of course for drug-specific precision. The intermediate precision that usually is the challenge using different, for example, equipment, different days. The run has been made by different analysts. Of course, linearity and range, and robustness, of course. Usually, our approach is to consider firstly to have a method set up in which a preliminary system suitability criteria and some parameters are defined, and test followed by a validation, whose steps are shown in this slide. Today, I really like to present a case study involving two bioassay kits developed by DiscoverX, which have been validated and used for release testing. First of all, we will see the results using the validation of GLP-1 bioassay. Just a brief look at the bioassay mechanism of action. As you can see in the slide, the glucagon-like peptide 1 receptor is a member of B family and G protein coupled receptor. It is involved in the activation of CMP signal pathway, and following a ligand stimulation, which in this case has been a client-specific drugs, GLP-1 receptor activity is monitored by measuring the cellular CMP levels using an homogeneous gain of signal competitive cell-based assay. So the increase in signal is directly proportional to the amount of CMP in the wells. In this slide, it's shown the results for our validation. And first of all, look at the left side of the slide. So first of all, we decide to have some system suitability criteria in order to be sure that each test that is run in our facility can be considered compliant. Then we look at linearity tests and, of course, in sample dilution. This is a critical process since these two parameters are really related to the nature of the client-specific drugs. So generally, we can start from a general approach, but then we need to be sure to have the specific range of linearity and sample dilution that needs to be applied specifically for the molecules that we used. After that, we look into accuracy tests. We run the precision. In each test, we check the specificity of the method, and then we confirm the robustness and the range of assay. And for the last part, we check the specificity of the molecules in this case. So we run just a placebo, and we need to obtain, in this case, no signal. In the right part of the slide, you can find the results, the practical results. So with number one, we check the positive control, and so it has been considered as one of our system suitability criteria. The second part was related to the linearity, and as you can see, we obtain very nice results for linearity part with an R2 parameters major than 0.99, and the sample dilution has been compliant to what has been expected. Overall, we check also the accuracy. In this case, as Gaurav said before, we check the 50% until the 150%. I have to be honest, in this case, this range can be varied depending on the nature of the sample that you need and, of course, on the client's requests. We then check the repeatability and intermediate precision. The intermediate precision has been checked using different analysts in different days and with different equipments, and we can obtain very nice results. The specificity has been confirmed, and also the test result is robust, and the range has been confirmed what has been set into the protocol. So overall, we can say that this full validation has been resulted compliant, but we will come back to an overall result after the presentation of the second case, which is represented by beta-arrestin and the validation GLP-2 bioassay. Also, in this case, we had a specific client molecules, but the mechanism of action is a little bit different. Indeed, glucagon-like peptide receptor is fused to a small beta-galactoside fragment and is co-expressed in cells that stably expressing a fusion protein of beta-arrestin 2 with a larger beta-galactoside fragment, as Gaurav presented before. Ligand binding activation of GLP-2 receptor stimulates the recruitment of beta-arrestin 2 to the cell surface receptor. This forces complementation of the two enzyme fragments, which, on addition of a specific substrate, results in a chemiluminescent signal that can be, of course, detected by a microplate reader. This assay provides a direct and universal platform for measuring receptor activation. Again, in this slide, you can find the result of our method validation. Also, in this case, we started defining different system suitability criteria and then going into linearity, sample dilution, accuracy, precision, specificity, robustness, range, and placebo. First of all, one of the most important criteria for system suitability is the reaction of positive control, and also in this case, you can see that the positive control result is compliant. And then we look at the linearity of both reference standard and GLP-2 agonist that in this case has been our molecule of interest. And also in this case, we obtained very nice results for R2 parameters and also for L parameters, ranging from 0.99 to 1, which is a very, very nice result. Then we challenge different sample dilution concentration, defining the range of treatment for the cells. And also in this case, we obtain a compliant result to what has been stated in our protocol. We test accuracy, and also in this case, you can see the results of accuracy test and the linearity test. In this case, the accuracy has been tested from 50% to 150%. The relative potency interval needs to be a little bit larger because this was the specification for our client. And as you can see, the reference standard and the theoretical trend are pretty the same line. Then here we look into the repeatability and intermediate precision. The repeatability was really nice with a coefficient variation of 9, and the intermediate precision was a little bit larger, but compliant with the validation criteria set by our clients, that is being related to the complex nature of the biological product, that sometimes can be not so homogeneous. Then we check the specificity, and also in this case, the needed result is specific and robust, and the range is being considered and demonstrated the same that has been stated in the protocol. We also check the specificity of the molecules, and as explained before, the reference standard and the positive control resulted compliant, whereas the placebo does not give any kind of signal as expected. So just to summarize both validation, as you can see, we reported in the left side a table which all parameters, and we can say that the specificity and the agonist parameter has been in both cases resulted compliant with 2 pass, and all the parameter challenged in this kind of validation, such as linearity, specificity, accuracy, precision, both repeatability and intermediate, and robustness, resulted compliant with what has been stated in the protocol, and sometimes with better results that we expected. So we can, of course, define that both methods show results to be accurate, precise, specific, and robust, and the CAMP anti-liraglutide bioassay kits will be used to determine the potency of some drug product to be released, measuring the cellular CAMP levels that in turn reflect, of course, GLP-1 receptor activation, and the parameter CHOK1 GLP-2R bioassay will be used to determine the potency of GLP-2 agonist for treating adult patients deficient for GLP-2R activation by monitoring, of course, beta-RS in recruitment as a readout of receptor activation. And these either output technologies represent useful tools to validate ad hoc bioassay that mimic drug mechanism of action. In conclusion, we can see that Eurofins Discoveryx offer a portfolio of GLP-1 and 2 cell lines and the ready-to-use bioassay for characterization through QC.release. And these cell-based mechanisms of action are reflecting functional assay, and we can say that are robust and sensitive, and evaluate both targets, GLP-1 and GLP-2 receptor, for a comprehensive understanding for therapeutic mechanism of action with different readouts. They can accelerate your drug release program into QC lot release. And of course, we can see that working together with us in Eurofins Bioproduct Testing Italy, we can provide support on assay validation in addition to supporting biopharmaceutical companies throughout the biologic life cycle, from analytical method development to market product release, analysis with drastically increase the capacity. I'm really happy to say thank you to Gaurav, which supports our work, and of course all people that work together with me and Gaurav to present this data today. Thank you for your attention.
Thank you, Gaurav and Julia, for your excellent presentations. We now have the pleasure to introduce our question and answer session today, where the speakers will answer all of your questions. Don't forget that you can still submit your question using the question panel in the menu on the right. So let's take a look at the first question today. So the first question that's been asked is, which statistical parameters need to keep your assay acceptance criteria for potency assays?
Thank you for these questions. First of all, most important parameters to be taken in consideration is the range of the potency. Typically for bioassay, the range starting from 80% to 125% respect to the reference standard. Moreover, you have to decide which statistical parameters do you want to use as a system suitability criteria for the test. In this specific case, we had different system suitability criteria based on statistical analysis. One of the most important is the slope ratio. So you need to calculate the ratio between the parameter B from reference standard and your sample, and typically this range starting from 0.8 to 1.25, it's a very nice ratio, but take into account that sometimes can also be different numbers on the basis on the nature of the molecules and the performance of the assay. Moreover, you have to demonstrate the parallelism between the standard and the reference the sample, and you have to calculate the coefficient variation between your replicates and for the parameter specifically for intermediate precision and repeatability. Usually a very nice CV to use for bioassays is minus than 20%. Taking account that bioassays are very, very challenging and sometimes very variable, so these parameters can also be different from a situation to a situation. Sometimes it's also useful to add the non-parallelism test, the F-test, and other statistical tests that are depend on the nature of your assay, the nature of what do you will use your assay to do, for example, for release testing, for a research phase, and so on.
Thank you. My second question is, how many data points in the standard curve are to be considered for parallelism?
Thanks again. Also, this question is very interesting. The ICH advised to use at least 5 linear points, but to be honest, we use a larger curve. We prefer to have 2 asymptotes, one lower and one upper, so we have usually at least 6 or 7 points linear in the in the exponential phase, and then we have at least 3 points for the upper and 3 points for the lower.
Great, thank you. And do you use phosphodiesterase inhibitors in your assays? And if so, which one and what concentrations do you use?
Right, I can answer this one. So for the GLP-1 assay, the CAMP assay, we do not use IBMX or any phosphodiesterase inhibitors. I mean, customers have used it. There is not significant difference between the two. So during optimization, we do look at IBMX and really don't see much difference between the two. The stimulation is only for 30 minutes, so it's not too long to really worry about the PD chewing up the cyclic AMP. So typically we do not recommend using that, but I mean, if your process of your protocol really needs that, you can definitely try that in your experiments.
Thank you. And what is the cell line used for the GLP-1 bioassay?
Right, it's GLP-1 cells that we use. This is a stable cell line from which the bioassay we derived. GLP is an interesting cell line. It has good expression for adenylyl cyclases, so that's a key reason we use GLP-1 cells for cyclic AMP assays. Usually, you have much nice activity for cyclic AMP in this particular background versus, for example, HEC293, which are not known to have good adenylyl cyclase expression in them, endogenous expression. So GLP-1s are our choice of cell lines for building cyclic AMP assays.
Great, thanks, Gaurav. And what would you say are the weaknesses of the EFC assay?
Okay, that's a good question. I mean, of course, you have seen part of the presentation I talked about that we created 10 plus different MOAs from EFC assays, how we use our tags for EFC intelligently on different proteins in the cells, and created so many different kind of assays. So I think, yeah, definitely there's always some things you need to be cautious about. It's a chemiluminescence assay. So one thing we recommend customers to look at, if the compounds of the molecules that they are going to add into the assay is not too colorful, that can quench the signal. So that's something that to keep in mind, that if it is very orangish or yellowish or something very extreme color, can quench some of the signal when you are, it's a top read assay, you are reading in a chemiluminosity. So that is something to keep in mind. Colorful substances can inhibit. But I think by and large, 99% of the times or even higher, we don't have this issue. Particularly small molecules coming into some kind of a unique color or something can do that, but typically for biologics, it's never been an issue. The other minor point is that if for some reason your molecule somehow inhibits beta-gal activity itself, that would be a red flag, because it's a beta-gal enzyme assay. So rarely we have seen at times that somehow they have an off-target effect on beta-gal. Those are the things that can inhibit, but other than that, it's pretty able technology. We don't have any other downsides that I can kind of really say for EFCs. We have built 1,000+ assays based on this technology, so it's pretty versatile.
Thank you. And have you done any NAB assays for GLP-1 and GLP-2 drugs?
Sorry, can you repeat that one?
Yes. Have you done any NAB assays for the GLP-1 and GLP-2 drugs?
NAB assays, yes. So I think we have not done ourselves at Eurofins DiscoverX. Of course, we don't have access to NAB samples. I know for GLP-1, customers are using GLP-1 assay for a NAB assay. GLP-2, I have not seen that, but I mean, the assays are built so that you can use for potency as well as NAB testing. We do have serum tolerance data for GLP-1, and that looks very promising. You can actually add very high concentration of serum and still does not inhibit the assay. So that also feeds into the first question about the EFC, what to be careful about or downside of EFC. So on the other hand side, it's upside of EFC that doesn't mind serum into the assays, so it can tolerate high levels of serum, and that makes our assays really good for building NAB assays. So we have used up to 100% of sample volume into the assay, just dumping entire ligand volume into the assay as pure serum, and still it didn't inhibit the activity that you get from the assays. So yeah, I mean, really good assays to build NAB assays based on serum toleration.
Thank you. And can your GIP receptor bioassay for GLP-1 bioassay compare ligand activation for dual agonists such as tirzepatide?
Excellent question, and I know I'm answering most of it, but yeah, this has been a really point of interest for us, a focus for us to develop an assay. Of course, if you're looking at the activity for GIP and GLP separately, you can definitely use this assay as a preliminary thing, your molecule is hitting GLP or not, and that's what people are doing. It's a new drug on the market, fairly new, I will say, and we are trying to develop a dual kind of an assay if possible to look at this molecule. Is a lot of interest, just like semaglutide. It is involved in obesity and diabetes, metabolism control and things like that. So it's very high interest for us. But yeah, if you are looking just for GLP-1, you can use this assay, but you need to be a little bit more patient for us to come up with a dedicated assay for this molecule, and we are working on it.
Great. And is the F-test mandatory to be included in system suitability and assay acceptance criteria?
Julia?
Yes, nice questions. First of all, it's not mandatory, since for the bioassay there are very, very few mandatory things. I attached some reference, such as Pharmacopoeia, USP Pharmacopoeia, that have some indication, but it's not mandatory at the moment. To be honest, it's useful to use not for system suitability, but for sample suitability. I mean, to understand if the specific sample within the test can be considered valid or not. But if the question is it mandatory? No.
Great, thank you. And if you would compare Cisbio HTRF assay or Promega Camp Glow assay with your assay, how sensitive is your assay or similar?
Excellent question, and this is very mindful of us as a company who develop assays for a living to keep in mind what competitors are doing. And I will not go into details, but in short, they are very sensitive as compared to HTRF, which is kind of we really see industry using this Cisbio assay. Promega's assay, we have not seen high penetration in industry, but I mean, these assays that we have developed are very sensitive and more sensitive as compared to Cisbio. But that's just not the only thing that is sensitive. They're also resilient. The way they produces the signal is a gain of signal assay with our kits. So you do get a gain of signal response, as Julia has shown, with GS coupled receptors. So that's a good thing. So your data analysis is much simpler. You are not kind of getting an activity which is loss of signal and you convert it out. And due to that, you also get a very high signal to background too, because they're simple gain of signal assays, easy to perform. You don't need any fancy equipment to read our assays. Those are some of the minor things. But more or less, I will say, yes, the assays are sensitive and they're gain of function, producing much robust windows. So typically you will see with Cisbio assays, the assay window is not that large, and that becomes an issue when you are doing something what Julia has been showing about validating assays and things like that, where you need to start with a robust assay window. So those things become a little bit of bottleneck if you really look into the robustness of assays. And with our assays, you see it really opens up. At times you see 10, 20, 30 fold windows with in cyclic AMP. And that's what you need to start in your potency testing, to have a reasonably large assay window, robust assays and sensitive assays, to be able to get that performance across days, across months, across plates, across analysis and things like that. So those are some of the things that we have dialed in on our assays. And again, there are some other things you can do, modulate how you are using your samples. There is a step that you can do if your sample has high serum or high concentration of some buffer which is not suitable for detection. You can replace that in between. So those are some of the things you can do with our assays. So they are more flexible, so to speak, to actually run compared to other competitors like Cisbio and Promega.
Thank you. Does your assay differentiate the GLP-1R and GC-GR activation? And guessing you need 2 different cell lines for it.
Yes, they can definitely. There are specific assays. They are not. They do not cross-react. You'll see a clear difference between the two. So specificity is there, which is kind of a hallmark for an assay to be in GMP space, that it's specific to the molecule it is activating. But good question. We do check those things overall with our assays, that they are specific to the molecule, specific to the pathway, and things like that.
Thanks, Gaurav. So Julia, a question for you. In which drug development phase is it possible to use these kit applications?
Thank you for the nice questions. I have to say that for our experience, we used this kind of kit both for preclinical studies, clinical development, and market approval phase. you can adapt the qualification, the validation, and the experiments that you want to perform on the basis of your needs. Of course, we are a GMP facility, so we are experienced for the clinical phase and for the QC release phase. But as I said before, we also use this kind of kit in the R&D phase, also in order to maybe compare different formulation of sample and to check which one is the best to use in this case, especially for the potency. So they are very, very challenging and useful in all these three phases. I'm sure that also Discoverex can also offer some specific other models, maybe useful more for the discovery phase, but these specific kits are more useful for preclinical, clinical, and QC release.
Thank you, Giulia. And for Gaurav, what are your risk mitigation approaches for long-term programs?
Right. Yeah, this is a good one. And I did kind of mention in one of the slides, didn't go into much details, but we do have certain, I would say, contingencies to make sure that we are providing long-term support, long-term supply of our kits, and risk mitigation as well. I mean, key of them are we do offer cell banking to customers. We can create a dedicated master and working cell bank for customers in which we kind of put them in place, and every time we build a bioassay lot, we'll go to the dedicated cell bank and produce it from there. That kind of reduces a lot of burden on bridging studies, lot bridging. So that kind of is very helpful to customers to not kind of do extensive bridging between lots. So that's one place we definitely do. Second one I will say is we do store material at 2 different sites for these banks as well. So that also is a big step in risk mitigation. It's not only one site. If something were to happen, we have the material safely stored at other site. And this is also a very kind of key requirement for a lot of programs that we support, is that we have this particular risk mitigation in place. And of course, some minor things like we do take care of the QC process that we do is a high level of manufacturing consistency we bring in, and that ensures a good quality of lots, consistent quality of lots that goes in year over year. And how we QC is important, because how lots align to each other is pretty important for us, because you buy a kit on year 1, buy a kit in year 10, they have to align really well. So those kind of strategies we have in place to make sure that we are within a very tight range when we supply kits to the customers. So between years, across years, you can really think of the consistencies maintained using our kits.
Thank you. And how can you assure an uninterrupted supply for kits, since a commercial release will need these assays for over a decade?
Okay, sorry, can you repeat the question? I just wanted to see if I missed one part of it.
Yes, of course. So how can you ensure that an uninterrupted supply for kits, since a commercial release will need these assays for over a decade?
A good question. So yes, I mean, that's kind of a cornerstone for us, and for any program that goes around 15-20 years easy for potency testing, we have to be very mindful that the supply for the kits are uninterrupted. We do manufacture and supply them, and make sure that the sponsor, the program is not affected. So there are a couple of things we do. We do sign supply agreements in which we can make sure that we are committed to manufacturing the material year over year. That's one of the key part that we put in place. And other than that, of course, creating cell banks, as I mentioned, is an important part where we ensure a reliable supply for bioassay cells is maintained throughout years. So those are a couple of things that we ensure for making kits available across years. So definitely there are a lot of other things we can do depending on the program to make sure that we can build larger lots for them if that were the case. So you don't have to buy kits every time from a different lot. So we can create a large lot, do reservations, do storage, and things like that.
And also, really, any large they want the trends that we take it, so we need to shoot that anyway. And then the only one would.
Okay,
Sorry.
thank you. It's okay. So Giulia, how long would you say a validation process takes?
Very good questions. As I said before in the presentation, we always apply a setup phase before to go into validation in order to check how the drugs respond to the assay, because these assays are very easy to use, but you always need to take into account that the molecules are very complex, so sometimes you have to check before to go into the validation how you want to perform the assay, and sometimes we have to also adapt the concentration of the drugs that you want to give to the cells, and so you have to check these things before to go into validation. So usually starting from setup phase to the end of validation phase, considering all the process and not only the analytical activity, but also the documental activity that is really important when you want to validate a test. For a normal, a classical bioassay validation, you need to consider at least 4 months.
Great, thank you.
And it depends also on how many runs you want to do for each parameters, okay? Because there is not a specific or a mandatory rules, but you have to adapt on the basis on your needs, and of course on the basis on how many batches of the sample you want to validate, because sometimes we perform validation on 3 different batches, and so in this case the validation process takes more time, or sometimes we use just 1 batch.
Thank you, Julia. And as a final question today, so terzepeptide is marketed at 6 dose concentrations. So what are your views towards developing the CAMP bioassay using a particular dose of Manjaro for reference, or would you use all doses separately?
Good question, and I know it could be Julia's question too, but I mean, we have to really see this particular one. I mean, development-wise, it will follow the guidelines that we have in place, like we did for semaglutide. Julia showed data for that too. So, I mean, this goes into our strategy for developing assays, and we will do a range from 50 to 150 or at times 200% into that. And I think everything else remains the same, probably in terms of how the curves will look like and the similarity and things like that. So, I mean, essential part is that there's nothing different in terms of it. It's just that we would like to develop an assay in which we can record the activity of both GIP and GLP, because that's what the molecule is. Manjaro, it targets both of them together. So, but development wise and qualification wise, it will follow the same ICH guidelines that we have for developing other bioassays.
Yeah, maybe I can add and confirm what Gaurav said. It's not useful to have just 1 dose. You always have a range of doses, and take into account that the sample and the standard needs to have the same dose in order to be compared.
Right.
Great, thank you both. So unfortunately, that's all we have time for today. So please join me in giving a great thank you to Gaurav and Julia for their time today. As you leave the webinar, a survey will appear on your screen asking you to rate the webinar. Please take a moment to provide your feedback. If now's not a good time, the survey will be sent to you shortly via email. And if you could complete this when you could, we'd greatly appreciate it. On behalf of Drug Target Review and Eurofins Discover X products, I'd like to thank you all for attending today's webinar. We hope to see you again soon. Goodbye and thank you.
Thank you.
Thank you. Bye.